So my talk is going to be about discovering genetic associations, and in particular doing so on large data. So when it comes to personalized medicine, one of the key things we want to be able to do is to find these associations between genetic markers and traits of interest. In the case of medicine, some of the obvious traits that we're going to be interested in looking at is whether or not you're going to get a disease or the propensity for you getting a disease. We want to consider whether you're going to react badly to a drug or react favorably to a drug. And the more we look at these problems, finding associations between genetic markers and traits, the more we find that, at least for common diseases, these associations are very weak. There's usually lots of associations, or there's associations between lots of genetic markers and a given trait, but each particular association is very weak. So in order to find these associations, we need lots and lots of data. Now, fortunately, we are getting lots and lots of data because, as probably most of you know, there's an exponential decrease in the cost of sequencing the genome that's happening. So a little bit over a decade ago, it cost $100 million to sequence the first human genome, and now we're about to see whole human genome sequenced for under $1,000 a person. And in addition, right now, for under $100, you can measure easily 1 million genetic markers in an individual. So that revolution that exponential decrease in the cost of sequencing DNA is leading to this wealth of data that, at least in principle, can help us find these very weak signals. But now the issue is, how are we going to do it? So one of the common ways of looking for associations is what's called the Genome-Wide association study. And what you do is you collect data for as many individuals as you practically can. You collect data on individuals who have some trait of interest. For example, they might have some disease. And you collect more data on other individuals who don't have that trait. For example, they don't have the disease. And then you measure, let's say, 1 million or more genetic markers for each individual, and then you look for differences between differences in the genetic markers between the two groups of people. And the map that I show here, each one of these bars, vertical bars, is one of your chromosomes. I think they're labeled 1, 2, 3, and so forth from left to right, top to bottom. And each colored dot next to a particular position on a chromosome corresponds to some interesting finding. So some interesting association between a marker on that part of the genome and some particular trait of interest. So as you can see, there are many, many associations that have been found. And in fact, this slide is rather old, and there are many more than what's shown on this slide. And you've probably heard, driving on the radio, probably every week, some announcement of some new finding of this sort, some association between your genetics and some trait of interest. Now, just to give you an example of something that we've done in my research group, we took about 500 people from Finland that have Lou Gehrig's disease, otherwise known as ALS, and another 500 people who didn't have the disease. And we did this search for differences across the genome. And we found a few markers in a particular region that had a very strong association with whether or not you had Lou Gehrig's disease. And then Brian Traynor at NIH, it took him a couple of years, but once we knew there was a clue that there was a genetic marker that was in association with Lou Gehrig's disease, he went in and in detail looked for what was the actual genetic change that was responsible for Lou Gehrig's disease. And he found it, and we now understand that this change accounts for about a third of all familial Lou Gehrig's disease, at least in Europe. So this gives you an idea of sort of the exciting things you can do with these genome-wide association studies. Now, these studies have been going on for several years now, and all the obvious, very strong signals have been found. Maybe not all, but a good number of them. And so most of the remaining signal is weak, as I had mentioned. And so we need, again, as I mentioned, lots of data to find these signals. And various groups are collecting very large data sets. Just to give you an idea of how large they are, groups like Decode, 23andMe, and Kaiser have all collected over 100,000, a large set of genomic markers for over 100,000 people. Okay, so now when you're in this situation where you're collecting lots and lots of data, what often happens, as Julie mentioned at the beginning of this session, you get what's called confounding. So you do a rather straightforward statistical analysis, and you find a very strong association between some genetic marker and a trait of interest. And it turns out that that association is false. It looks real when you do the statistics, but it's actually not real. And what's going on is what's called confounding. Examples of confounding are you have in your data people with multiple ethnicities, or you have closely related individuals, and because of this, you get these false signals. And let me try to give you some idea of how this happens. Let's just consider a single genetic marker now, which can either be an A or a T. These are the nucleotides. And we're looking at one particular, let's say, disease. And in this figure here on the left are the people who represent people who have the disease, and on the right are people who don't have the disease, the so-called controls. So if you look at this data naively, you see, the cases, there's more A's than T's. Each circle here corresponds to a particular individual. And then if you look at the controls, you see, there's more T's than A's. So you think you found an association between this genetic marker and whether they have the disease or not. But it turns out this is a false association for the following reason. It turns out that in this data, there's actually two populations of people. You can think about them as different ethnicities. And in population 1, there's more A's than T's. And in population 2, there's more T's than A's. And it just so happens that in the cases, there's more people from population 1, and in the controls, there's more people from population 2. So that gives you a false signal. And so what we want to do is avoid this and yet use lots of data, for which this sort of confounding is almost inevitable. So how do we do that? Well, many decades ago, animal breeders had the same problem, and they figured out how to solve the problem, at least in principle. They developed a statistic model called linear mixed models. These models are great for finding these hidden confounders and getting rid of them. But there's a catch, and that is they're very computationally expensive. So for those of you that know computer science, if you're analyzing n individuals, the runtime will be O(n) cubed, and the memory use will be O(n) squared. For those of you that aren't computer scientists, just appreciate that once you get over about 5,000 individuals that you're trying to analyze, you can't do it with this algorithm. But what we've done in my group is to figure out with some basically algebraic tricks how to bring down these heavy computations to very simple computations and not sacrifice the accuracy of the result. In fact, what we were able to do was show that using the tricks that we developed, not only were you able to make the algorithm run faster, but you actually got more accurate results than you did if you used it the traditional way. And that's a very rare thing that happens in computer science. Usually when you're clever enough to think about how to speed up an algorithm, you usually introduce some sort of approximation, or you sacrifice the accuracy of the results somehow. In this case, it actually was the opposite. By running faster, we were also able to run more accurately. So we published this work in Nature Methods in 2 different publications. This is joint work with Jennifer Lisgarten and Christoph Lippert. And we are now taking this algorithm, and because we were able to speed up the analysis so much, we're now tackling problems that are very difficult. And here's an example of one. There's some data from the Wellcome Trust for 7 common diseases, including coronary artery disease, hypertension, type 1 diabetes, type 2 diabetes, and so on. They have lots of individuals in that data set. There's about 15,000 individuals. And what we were able to do is analyze, not only look for associations between single genetic markers and whether or not you have this disease or not. But we were able to look at interactions between these genetic markers, what are sometimes called SNPs, or single nucleotide polymorphisms. So there, if you look at the markers that were measured in this Wellcome Trust data, there's about 60 billion pairs of them. So we asked the question, for every one of these 60 billion pairs, we asked the question, is there an interaction between these SNPs and whether or not you get this disease? So because of FaST-LMM and because we have a nice parallel computation environment at Microsoft known as Azure, we were able to do these computations in a relatively short period of time. If you were to do it on a single machine, it would take 400 years, but we did it, in a day or so. Sure enough, we found some new interesting biological interactions in this data, which unfortunately I don't have time to talk about today. So in summary, when you're looking for these associations, it's good to have a lot of data because a lot of these associations can be very weak, at least when you look at them one SNP at a time or one genetic marker at a time. But there is an algorithm that can handle this. And when you have lots of data, you have this very large possibility of confounding. And fortunately, there's an algorithm that can handle this confounding problem, namely linear mixed models. But now we've made them practical to use. And if anyone out there is interested in trying it, it's available free for academic use. Just search for FaST-LMM and you'll find it. Thank you very much.